name is Chun Meng. I come from Shanghai Veterinary Research Institute, Chinese Academy of Agricultural Science. In China, only 3 national research institutes, a veterinary research institute. Another 2 is located in Harbin and Lanzhou. Our institute is located in Shanghai. My name, I want to give a simple introduction. Meng is my family name. Chun is my given name. Chun in Chinese means spring. I really was born in spring, so my parents give me this name. So this is my first. This is my first time being to Pakistan. First, I thank Dr. Zepp's invitation. I really the delicious food and the kindly and warmly of people really give me a deep impression. So I really thank the organizer. Okay, your dedication impress all, and also to the audience. Let's shape the future together. This is my outline. First, 4 sections. First is the overview of the ND and NDV. Second, about the brief introduction of Class 1 NDV. First, what is Newcastle disease? It is one of the noticeable diseases of OIE. It can cause big economic losses and remain a major threat to the poultry industry. And the cause agent is NDV, which is the enveloped RNA virus belonging to the family of paramyxoviridae. And it can cause high morbidity and mortality in poorly vaccinated chickens. Even can cause high mortality in pigeon and domestic waterfowl, such as duck and goose. And in the well-vaccinated layers, it can cause dramatically drops in egg production. So this disease ranks the 8th of the animal disease, including all the livestock and poultry disease. And the second most significant poultry disease, just behind the avian influenza virus. So in the past 10 years, 109 OIE members report the disease, such as we can say almost all the country. There is one question I also want to discuss with our colleagues, because we know in the world, the 3 countries produce many chickens: China, USA, and Brazil. I already searched from PubMed. There is almost no research paper from the Brazil about ND. And also in OIE data, there's no ND outbreak in Brazil. I don't know why, because it is population is huge. Also, the environment is also like the southeast. It's very easy to culture virus. So I don't know why. If you anyone know the answer, you can contact me. The first outbreak ND is in Newcastle, UK and Indonesia, almost at the same time in 1926. This already exists 5 or 4 pathogenic pathotic. No, we're still in the 4th from the 1980s. It is genotype 7, which is responsible for this outbreak. But some people should divide it to the 2 outbreak, because from the 2000s, the genotype 7 can divide it to sub-lineage. But in my opinion, I think still belong to genotype 7. So etiology of the NDV has changed. His family belongs to the paramyxoviridae. The subfamily is the Avilaria, and the genus in how to say is also Avilavirus is the new genus for NDV, and the species is the avian also Avilavirus. In previous, we call it APMV as a brief, and also AOAV but we never use AAVV later because it already discarded this classification. So this is the genome structure. It contains a single strand, negative sense, and a non-segment genome about 15.2 kb. It contains 6 gene order from NPP, M, FHL, and it can totally 6 gene can produce 8 protein because the P gene can through the RNA editing can produce the V protein and a W protein. The fusion protein is very important because it is the determinant of the virus pathogenicity because its fusion is the first step to ensure the virus can infection. This is a simple introduction because of time limited. I will omit this. So as we know, the OIE, how to say, divided the NDV to 4 types: aberrant and letogenic and pathogenic and velogenic according to ICPI. This is explained by the 1-day-old SPF chicken injection. But IBDT and also will be considered. So the diversity of NDV can be very huge because it can be class 1 is 1 genotype and in class 2 or to know it's 21 genotypes and the host very diversity. No matter the domestic bird or the wild bird, more than 200 species can infect it. So this virus, like even influenza, never can be animated by human because the hosts are too much. This I will omit because time is limited. So this is the class 1 NDV. The unique character of class 1 and class 2 is that its genome length. In 2003, with people first think that exists class 1 because it's a genome about 15189 BP is about 12 BP insertion under the P gene. And some character of class 1 is in my personal opinion that first, it's first isolated from the wild bird, but no, more and more isolated in domestic poultry. Second, all the nature isolated belong to aberrant, only except one, airline strain. Third, it's most important that some class 1 strain are easily to mutate to the virulent one in short time. These are 2 examples. First, that the name, the strain name we are just through 9 times and 9 times in chick air sac and 5 times in brain. It generated the virulent strain from the ICPI to equal to 0 to ICPI to 1.7. Another one is which down our life that GS10 just passage in chick air sac for 10 times. The ICPI increased to 1.9. So this is the data from China that no, in China, more and more class 1 NDV can be isolated in domestic poultry, no matter the chicken, duck, and even the goose and pigeon. So it's cost totally transferred from the wild bird to the domestic poultry. And also we can say, this PPT is some problem. This is the data from the China Animal Health and Epidemiology Center. Also OIE reference laboratory of ND that in the 2014-2019, total 1,432 NDV strains isolated, but more than 1,097 belong to class 1. Only 335 belong to class 2. It's very, very strangely, I think. So here I found some unique picture of NDV. This is the paper already published that we how to say, design a very simple experiment to verify why class 1 NDV can easily mutate to the variant one. Because first we should know this is the poultry species, means everything, even the gold, never 100% pure. It can contain some other things. So also the virus, even we cross the blood purification by 3 times or 5 times, it never can be very pure. So it's very, it contains a mixture of the virus. Because we know in the for evolution of NDV, there's a good example in Australia. Australia is a lonely land, far from any other country. But in past 60 years, Australia exists the no NDV and exists the aberrant NDV. Then exists the variant NDV. So it's a very good example to demonstrate the how NDV evolution. So, but the main point is about F0 cleavage site from the same virus in Australia, just directly mutated from the variant to the variant. So we have 2 questions. What about the evolution of co-species in class 1 NDV during the virus evolution? Okay. And also there are different pathogenicity between the live-generated variant NDV and the nature classical strain. I think, okay, I give you just, because time, launch time is coming, I will save our time. This is, we use the deep sequence method to, how to say, there is a one same strain through 2-way passage. One is through the chicken air sac, another through embryo. Through the chicken air sac, only 10 times it becomes the variant because it clear CPE. But in passage through the embryos, 20 times, no change. So we want to know why. We use the deep sequence, and this is the sample we want to determine the GS10 P means the plasmid, because plasmid sequence is fixed, never be changed. So after the sequence, we get so many data. Each strain about more than 20,000. It can be statistical. And also we found that why GS10 was easily mutated to variant, because it is some variant cleavage side mutant dramatically increased. This is with some category of the method of statistic, and also this is the kinetics. So after passage, the virulent post-space increased dramatically. At the same time, the aberrant post-space decreased dramatically. But during the passage in the embryos, no change. Here is our data. And also we found some unique character for this NDV. The man, this is the man data, means this artificial genetic generated variant NDV through nature infection way, such as eye drop and nasal drop, no deaths. It is very, very unique, but through the injection will be died within 5 days. So we do a lot of experiments to try to explore why. This is our experiment about, so in CPE, it's variant, but after nature infection, the tissue damage, no. And also this is a very tighter, how to say, is in nature infection, very, very low. But if you inject by venous and the muscular, it causes very quickly die. And also the virus shedding are different between this classical variant strain. So, and also we do another experiment. And also we do the receptor binding assay. So, how to say, and also some cytokine expression level after nature infection. No, because So this is our conclusion that GS10 air cytokine can rapidly induce production of high level of cytokines, but usually 10 to 100 times higher than first 33. So so we know the OIE standard for determining this NDV is belong to variant or variant only by 3 index: ICPI, IVPI, and MDT, but never do mimic natural infection rates. So its standard will be modified on, I think is a question, because in nature, no chicken infect NDV by injection, only by the eye or nasal contamination. So it's our question. So this is some, because Dr. Zeb said we should introduce some experience in control genotype 7 NDV in China. So this is the data from the government that really the NDV in China decreased dramatically from past 20 years. And also the poultry farm, which positive in NDV, is also decreased. This is the data from the China Animal Health and Epidemiology Center. And we know the strategy for disease prevention can be 3 departments. And I think the most important is the prevention. How to prevention good is the effective vaccine. Here, an effective vaccine not only provide the clinical protection, but also can reduce virus shedding. So we know this is a very normal picture in poultry farm. What virus shedding means what? One chicken share the virus, another chicken will be infected. Maybe in this poultry farm, the virus can be passaged through the chicken easily 200 times, because the nature passage, it can increase the virus pathogenicity. So how to decrease the virus shedding, I think is a good vaccine should be do. This is my master thesis about 16 years ago. My supervisor is Professor Xu Fanliu. He asked me to generation of vaccine candidate of genotype 7, but by reverse genetic and to evaluate the protective effective. This is our, how to say, totally Professor Xu Fanliu choose at least 3 candidates, vaccine candidates. This is the number 2. At the final on market can be sold is the number 3 candidate. So just we China used this genotype 7 ND vaccine. It really decreased the virus shedding. We do a lot of experiments, no matter natural infection or just injected by muscular. It use the homology vaccine can really decrease the virus shedding. I think this is why China control genotype 7 vaccine in past 20 years, past 50 years, have achieved the successment. So thanks for your attention.